forget that. It is my pleasure to welcome everybody to the February session of the ForestConnect webinar series. My name's Peter Smallage, and I am the New York State Extension Forester, and one of the programs that I oversee is the ForestConnect program that includes, among other things, this monthly webinar series. And it's to your great benefit and my great comfort that we often have some outstanding speakers, as we do today, Mark Whitmore. And I've realized Mark is probably the most frequent repeat presenter that I have, to the point that he's worn out his headset. I had to buy him I'm buying him a new headset. But it works for today. And Mark, as many of you are familiar, and because you're here, you know he's going to be talking about insects, and one of the insects of passion of late is the Hemlock Woolly Adelgid. Mark's been working on this a lot for several years, and it's my pleasure to turn the floor over to Mark. So Mark, thank you for offering to present, and welcome back to the webinar series. So I'm going to mute my microphone and just hover in the background. So it's all yours. Thank you.
Okay, hover away, Pete. Okay, so I guess it's actually, this isn't an insect of late for me. I've actually been studying Adelgids for a really long time. Actually, that's Adelgids got me into the field of forest entomology. It was the Balsam Woolly Adelgid out west in Washington State, where I grew up. And so, I noticed there's a lot of people there that I've already spoken to. I've spoken with before. People have seen my presentations, and I'm going to try and focus this presentation not on the threat as much as the management, the options that we're looking at, and the research that's going into developing those options. With no further ado, I will start out with a nice picture of the Hemlock Woolly Adelgid. These are very fat and happy Adelgids here right next to Ithaca on Cayuga Lake. We got the Adelgid here in the Cayuga Lake area around 2008, and since that time, my research program has really been ramping up on the Hemlock Woolly Adelgid. This is a picture of the worldwide distribution of HWA and the hemlock species. The interesting thing is that all hemlocks harbor the Hemlock Woolly Adelgid. And I was coming from the Pacific Northwest. I was sort of scratching my head because I'd never seen it on Tsugae mertensiana, which is a mountain hemlock. And sure enough, I found one at low elevation recently, and it had HWA on it. So as far as I'm concerned, all hemlocks harbor HWA, but in the areas where it's native, which would be here in China, where there's 3 species of tsugae, in Japan, where there's 2 species of tsugae, and then the Pacific Northwest, where there's 2 species of tsugae, they don't kill the trees. Indeed, they're just a native insect, and they come and they go, and they're controlled by a number of factors. I think primary among them are the predators. So the work of Nathan Havel was really important, I think, in looking at the genetics of HWA. There was one question we had in particular, and that was, where did the eastern population that we got come from? And Nathan found that it was actually the southern Japanese biotype that we have in the eastern US. And another big question that he answered was people had been assuming that HWA was introduced to the Pacific Northwest, and Nathan showed that indeed the biotype in the Pacific Northwest is distinct from those in the Far East, in China and Japan. So why we don't have the Western biotype from Washington and Oregon, considering the amount of commerce that we have in the nursery trade in the East Coast, is beyond me. Another one of those mysteries of life. So this is the big scary life cycle picture, which I really like, because HWAs have a very interesting life cycle. Basically there are one, two, three, four, five, six, 7 separate adult forms of this insect. There are 1, 2, 3, 4 of them on spruce, which is the primary host, and 2 on, or 3 on the hemlock, which is the secondary host. So the interesting thing is, in the Pacific, in North America, we have yet to find any HWA on the primary host, and we call spruce the primary host because that's where there's sex. There's no sex on hemlock, and we have yet to find any spruce in North America that harbor the sexual generation, and that's easy to tell because here you see this is what we call the galliculae, the gall that's formed by the HWA on spruce. And I actually have one of the Japanese spruce right outside my office, and I look at it regularly when I want to get out of the office, and I've yet to find any galls on those trees. So the distribution of HWA on the East Coast is interesting. So the story begins, actually, most of the literature says 1957 or something like that in the Richmond area. Well, the story is actually not that, forget that, okay? So recent work, and Nathan Havel's work, has shown that it was actually probably introduced in the early 1900s into an arboretum in the Richmond, Virginia area. And it turns out at that point in time, Japanese gardens were all the rage, and so it probably came in on nursery stock from Japan. And of course, you can see in Richmond, it's not very close to any of the hemlock forests, and so it took a while for the populations to build. But the date 1957 actually comes from when it was found in the 70s, I believe, causing huge problems in the Virginia area. They went to the National Museum at the Smithsonian, and the first accession of HWA from Virginia actually comes from the late 50s. So that's where that date comes from. So suffice it to say that HWA was flopping around in the Piedmont here in the Richmond area, and then it got up into the Appalachian Mountains and started to go crazy. You see the dark line is the distribution of Eastern Hemlock in North America, and the pink is the current distribution county by county of Hemlock Woolly Adelgid. And thank you, Tom McAvoy, for putting this nice map together here. And it is recent because the big news is that in Nova Scotia this summer, they found HWA, and not just a little bit, but it actually has probably been there for 10 years or more, and it's already causing probably thousands of acres of mortality in southwestern Nova Scotia. But this is a good representation also because we have the population here in Michigan. So the life cycle basically in North America is pretty simple. This is the 2 generations per year. Everybody's probably seen this, and I just wanted to go through it again just to be sure everybody's up to date here. So in the summertime, it goes through a state of estivation, the Sistens generation here, and it looks like this on the upper left-hand corner. So it stays in the summertime like something that nothing wants to eat, tiny and black and skinny. And then in October, it begins to grow and fattens up, and then it, as you can see in here in the upper right hand corner, that's probably in January. They get nice and fat, and then when they get in here in February, March, they begin to lay eggs. Now the interesting thing is that it's a pretty plastic trait. It's actually, we found eggs this season. It was a very warm fall, and we actually found eggs showing up in December here in Ithaca, in a warm location, albeit, but and just in a couple. But it was really shocking to me to see that for the first time ever. And so the phenology of this insect is something that we're really following carefully because it's important in implementing the biological control. Let's see. Okay, so the eggs hatch, and when the eggs hatch, they turn into the first instar crawler, and that is the only dispersal stage. Here you can see a little tiny one down here. So if the Adelgid is 1 millimeter in size, the crawler is probably about, we measured a bunch of them. We got them about 0.2 millimeters, 0.25 millimeters, so pretty darn small and easily blown in the wind, but also they can get carried by animals like birds, or perhaps on the fur of a mammal, or a deer or something, a mouse, a squirrel. So they can get around. They're actually pretty quick, and they have those little legs, so they can scoot along. Long distance dispersal, though, I think really every indication in my mind is that it's birds that are perhaps the most important for really long distance dispersal, because the only two times I've actually, I think, found the first trees in a stand that have been infested have been on the branches of hemlocks right next to water, and only those lower branches near the water, not the branches further on the other side of the tree, which indicated to me that perhaps likely that a bird had come in, landed on those branches, and then visited the water and depositing the crawler on that branch. So the second generation, the Progrediens generation here, it starts in April, March, April. It all, of course, is temperature dependent, and they begin to develop. And this Progrediens actually settle on the same tissue that the Cystins generation was feeding on. So this tissue has already been somewhat degraded by the Cystins generation, and Progrediens generation won't be as fecund as the Cystin generation was. But the interesting thing is that the winged adults will then go off, and that's a density-dependent character, the formation of the winged adults. And when they go off looking for a spruce here in North America, of course, they just don't find anything, so they just die. So it's like having a little vacuum cleaner sitting there taking off those Adelgids. And so the Progrediens generation adults lay their eggs, lay the Cystins eggs, the eggs hatch, the crawlers come out, they find a place to settle down on the brand new twigs of the year, on the current year's twigs, and then they go into estivation. So that's the basic life cycle of Hemlock Woolly Adelgid in North America. There's a charismatic picture of an Adelgid. Of course, they're all females. There's no males in the population in North America. And here you can see the stylets here. Actually, the stylets, when they're feeding, they form a single tube. This is, of course, a dead one, and the stylets have curled up around it. There's 4 different stylets, mandibulary and maxillary stylets, that form a tube that they puncture into the twig tissue and feed. And here you can see these nice little pores here. This is where the waxy wool is formed on the body, on the dorsal surface of the body here. So these are little antennae here, the little legs. It's actually a really good picture of an Adelgid. So the impact on the tree, this is the important part right here. So the Adelgid actually, it doesn't feed on the needle. What it does is it inserts its stylets into the twigs near the base of the needle. So, and it feeds on the xylem ray parenchyma cells. So there's a profusion of xylem ray parenchyma near where the needle attaches to the twig, and that's where they prefer to settle. So what happens when the Adelgid actually settles, there you can see there's 1 million of them up there, not 1 million, okay, probably 100 of them or so on that one little twig. And what they do is, I like to call this sort of like the voodoo doll model. If you can imagine each one of those Adelgids with its stylet sticking into the twig, it's sort of like putting its needle into that twig. And every place that needle goes into the twig, the tree, it elicits, basically a generic wound response by the tree, which it's mobilizing the secondary chemistry of the tree to wall that off. And basically what it does is it clogs up the conductive tissue. And when it does clog the conductive tissue, the needles don't die immediately, but the bud fails to develop in the springtime. So that's the thing, is it kills the buds first. The needles will still be on the tree, so you'll still have a green tree, but with dead needles, with dead buds, it won't be able to produce new needles. And so gradually the tree is worn down because it can't produce more photosynthetic tissue. And on really good sites, the trees can persist for a long time. Up here in the north, I know I've seen sites where the Adelgid has been present for 20 years or more. The trees look not very good, but it's still, they're still hanging on, I think, because they're in really good sites, and they're strong trees, and they can take that repeated defoliation. Whereas on dry sites, in the south in particular, trees die very rapidly. 4 years is pretty much the norm in the south, whereas up here in the north, I rarely see that, but I have seen it on very, very dry sites near Seneca Lake, actually. I think the droughts actually exacerbate the impact as well. I know that some of my friends think that the rapid demise of the hemlocks in the Great Smoky Mountain National Park was helped along by the advent of the Hemlock Woolly Adelgid getting into the area and then having a big drought for the next few years. So the length of time it takes to kill a tree varies, I think, according to site, with the most mesic sites being the best. So this is what it looks like in an early infestation. This is right up here near Cayuga Lake. Healthy tree tissues makes healthy HWA, greater fecundity. And this is really important when you're thinking about raising predators, which I'm going to talk about a little bit later here. You get nice fresh infestations, and you get better predator food. The host tree tissues, though, decline rapidly over time. And the adelgids in them decline in their quality. And so if you're looking for predator food, you don't want to go to those places where it's been there a while, like this place, where HWA density is diminished on the older twigs. You see a couple here, but most of the good feeding sites have already been utilized by the earlier generations of the adelgid. And you'll see out in the back here, they prefer to go into the areas where there have been new shoots that have grown, and that's where you'll find them when you get into these areas where you've had adelgids for a while. And this is a late infestation. Here you got a little spittlebug here, where the buds have died and the needles gradually fall off over time, given their lifespan of 3, maybe 4 years naturally. So that's the life cycle of Hemlock Woolly Adelgid and its damage to the trees. And I put this in here because this is why we're particularly nervous in New York State. We have an amazing density of hemlock. It's actually the 3rd most common stem in New York State, right behind red maple and silver maple. And if you look here, we have amazing concentrations of them in the Adirondacks, along the south part of the Adirondacks and Tug Hill Plateau, but also it's pretty much common throughout in the Catskills, throughout the southern tier. And the thing I like to remind people is that even when you don't have a lot of hemlock around, it's still a really important species ecologically, because basically it forms an island of habitat. It's a foundation species, and that habitat that they form, basically it supports, it's essential for the lives of a myriad of species that depend on it. And you think about it, it's an easy concept. Basically, if you have a hardwood forest and you take all the ash trees out, you still have a hardwood forest functionally. Whereas if you have a hemlock forest and you take the hemlock out, it's a totally different game. And that's one of the most important ecological reasons. That's one of the most important reasons we're really concerned about hemlock in New York State. Here's a progression of its, oh, it's not working. I wonder what's going on. This was a really nice progression of its movement in New York State. Basically in the late 80s, it started here in the lower Hudson Valley, spread into Long Island. It's been moving up the Hudson Valley, and then we got it in the Finger Lakes area. We got it in Rochester and Buffalo on a nursery stock movement, which is one of the most common, I think, long distance dispersals besides birds. And let me see this, whoops. And this is the current distribution in the state. Here you can see it's taken its time. This is a really interesting difference between New York State, I think, and further to the south. In the south, its movement has been very rapid, and there's been lots of mortality, whereas here in New York State, having started here in the Hudson Valley, it's in the 80s, it's taken 20 years or more to get up here. And we first found it just this last summer, Prospect Mountain in the Adirondack Park. And this was a real wake-up call, because we've all been looking at the Adirondacks and the amazing concentration of hemlock in the Adirondacks, and getting very worried. And it was a very fortuitous event that David Orwig from Harvard Forest was up there geeking about, looking at some old-growth forest on Prospect Mountain, and casually noticed that, oh, there's Hemlock Woolly Adelgid there. Well, he happened to be with a colleague from the SUNY environmental, the SUNY ESF campus in Syracuse, who knew that there were no reports of Hemlock Woolly Adelgid in the Adirondacks, and immediately the light bulbs went off and he was on the phone. And that was the big excitement this summer in New York State, was that discovery there. But here you can see in the Finger Lakes it hasn't spread very far, and the interesting thing also is there are periods where it spreads rapidly and periods where it doesn't. Here you see 2009, 2015, it was a massive period of expansion, and I attribute that to the warmer temperatures, very mild winters. And being a skier, I can attest that it was really terrible skiing during that period. But then when we get the polar vortex and the colder winters, where the skiing is really good, it really slowed down the expansion of populations. I'm going to talk more about cold weather and HWA later here. So this is our kumbaya moment to think about the ecological impact and what it means to lose the hemlocks. And one of my friends and colleagues, Jan Nyrup, one day stated to me that, well, just behind climate change, the loss of the hemlocks is perhaps one of the biggest ecological disasters we're facing in New York State. And I agree with him on that account. Of course, you know, it's indirect impacts on the stream, keeping the streams cooler, and which is necessary for the reproduction of salmonids, which is a huge economic thing in New York State with all the fly fishing and such. And it's also important, you know, not just the direct shading, I think, but also when you walk into a hemlock stand in the summertime, it's amazingly cool. And I think that not only retards the melting of snow in springtime when there's no leaves on the hardwood forest trees, the snow melts really rapidly, but in the hardwood forests, it sticks around for a while and maintains the cool groundwater for a longer period of time. So let's basically restate the problem. Asexual reproduction is the big thing. They're all females. That's really important because all you need is one individual to start a whole new population. The reproductive potential can be high. I think that conservatively, maybe 5,000 potential progeny from one female in a year. You know, maybe less than that, but still, that's a pretty high reproductive potential. And I've seen the impact of that when you get high winter mortalities, and all of a sudden the populations, they just bounce right back like nothing had happened to them at all. On the East Coast, there's no native natural enemies, which is interesting because, you know, to think about it, it's been around here on the East Coast for a long time, and I'm sort of surprised that there haven't been more generalist native predators that haven't popped onto it. Of course, I see a syrphid fly here and there, and I see some, you know, some coccinellids, some ladybird beetles feeding on them, but definitely not in the numbers that are necessary to affect any kind of biological control. And it's sort of like they don't get it. They don't get that there's this huge food source there. I'm sort of surprised. I'll keep scratching my head about that one for a long time. But we have been working on classical biocontrol, and I'm going to focus on that later on here. And so far, there's been no documented resistance, but there has been some work that points to stands that we think might be resistant. There's work going on right now in Pennsylvania where they're trying to see trees that have survived the impact of Hemlock Woolly Adelgid, as well as some work that was done in northern New Jersey. And so the whole process now is to figure out, is this really resistance or was it just dumb luck? And so we're working on it. It's important. It's going to take a while, but if we can find resistance, that'll be really important for the survival of Eastern and Carolina hemlock across the landscape. So once this to recap, so what controls natural populations? What are our options, basically, when we're looking at trying to manage this insect? Host tree resistance controls natural populations. There's many factors. We don't really understand exactly what goes on, but we're looking at it, and it's important not to ignore. Abiotic factors, temperature, I'm going to talk about that. Biological controls also are important to predators. There are no parasitoids for Hemlock Woolly Adelgid, and and the pathogens are all generalists. So really, it's biological control predators, abiotic factors, cold temperatures, and perhaps tree resistance that are all added up together to control populations where it's native. It's interesting to go to the Pacific Northwest where it's native. And when I was growing up there, I didn't learn about the Hemlock Woolly Adelgid as being a problem. And indeed, it's really fun to go and now to study it and to watch populations grow. And it's sort of like a game of whack-a-mole in the Pacific Northwest, where you get populations that begin to grow and they acquire, when they first, you get these initiating populations, small spots on a couple of trees maybe, and when they first start, there won't be any predators, but gradually over a couple of years, the predators will find those populations. The predator populations will build, and the predator populations just get really intense. And then all of a sudden, Hemlock Woolly Adelgid disappears. It very much like whack-a-mole, and I look at that and I think that really is I think the predator-driven system, much more so than host tree resistance. But maybe when you get into the Cascades at higher elevation, there are other factors involved, because I don't really find a lot of Adelgids up in the higher elevations of the Cascades. So one of the most important drivers, I think, of Hemlock Woolly Adelgid in the north, and why we're so different than the east than in the southern states, is the cold temperatures. And this is a map of New York State with the coldest temperatures through 2014, which was part of the polar vortex. And so I've been looking at two study sites, Minekill State Park and Teganic State Park. And Minekill still is perhaps the coldest place where we have HWA, and Teganic, it's pretty warm. It's in the banana belt sort of the Lake Ontario plain. It's on Cayuga Lake near Ithaca. And the interesting thing is that, so we've been studying mortality at these sites for a number of years now. And one of the things that we're finding is that every year is different, which, sort of, yeah, right, of course every year is different. But the reaction of Adelgids to the different cold temperatures is really remarkable. Here, this was 15-16, and here you can see the mortality was really low in the first part of the season, and then we got to St. Valentine's Day, and it was huge. We got down to about minus 23 degrees all of a sudden, and we got huge mortality here, almost up to 96, 98%. The interesting thing is that this was just the huge impact of cold temperatures here was because of this area right here. It was in the 50s for a couple of weeks before that drop, and we think that was a response. That was an acclimation phase where sort of the Adelges, they just weren't acclimated to the cold temperatures, and they were very susceptible. Whereas you get a more normal year here where you get colder temperatures, albeit not down to minus 20, but colder temperatures, and the mortality just sort of stays at very low levels all throughout the winter. 16-17 was huge. The populations, after 3 years of very heavy mortality, the previous 3 years, the populations just bounced right back in early 2000 and in fall 2017 and 18. I thought I had pretty much extirpated the HWA from the Minekill study site, and I went back in fall, and they were just all over the place. Plenty of bugs to work with. So this is another way to look at it. Here you get the mortality events occurring at different times of the year. Here you have an early mortality event in early January, and the two study sites are somewhat mimicking each other. Here you get a mortality event in February. This is the St. Valentine's Day massacre. But here you see Teganic was much lower than Minekill, and then all of a sudden they went way up high. Didn't kill them all. Populations bounced right back. And here you got 16-17, where the populations, the mortality was relatively low until late in the season, and it was this little kicker here in late April that really sort of surprised us with that mortality. So the interesting thing is when you think about the mortality and then you think about the predators. I've been having a problem thinking about the establishment of Laricobius nigrinus up here in the north, and how it's not really mimicking that which I see occur with population buildup in the south. And I'm looking squarely at this winter mortality as one of the, perhaps one of the drivers of their population buildup of Laricobius nigrinus. And I'll get into that, but here, just think about it. If you have all your prey die early in the season here, you're probably not going to have all the food you need to lay a full complement of eggs in the springtime. Whereas perhaps if you have the late die-off of the prey, then you might have had consumed enough prey to have your ovaries full of eggs and be able to build the populations. So this is something we're thinking about now. I'm thinking about building a model on that. But one of the things I think that's really important is that we don't consider the cold mortality as the key to the silver bullet, I could say, for controlling Hemlock Woolly Adelgid. And I got to go back and remind everybody about the Balsam Woolly Adelgid, which is one of my most favorite insects. And it's been wiping out the true furs on the West Coast, and now balsam fir on the East Coast. But when I first started studying Adelgids, back, I guess, around 79, 80, I tried to get money to study the insect, and I was told in no uncertain terms that it would never be a problem in the colder areas where true furs are important, because the cold temperatures would kill it off. It would be, the distribution would remain restricted to the areas proximal to the oceans, like Puget Sound and the Maritimes. But now it's a totally different thing. I've found it up near tree line on Mount Washington, which is one of the coldest places on the East Coast. We're getting large amounts of mortality now in the Lake Placid area, which is high in the Adirondacks, very cold. So if the Balsam Woolly Adelgid can adapt to cold temperatures, I think that my bet is that the Hemlock Woolly Adelgid will as well. But I'm incredibly conservative when it comes to conservation of a species like hemlocks. And so if I'm wrong, that'd be great, but I'm going to act on the information that I think that it can develop, and that would be losing time if we didn't effect management at this point in time. So I think one of the most important things that we need to do, that we can do right now, is implement biological control. We're going to, by implementing biological control, it's basically a classic biological control project where you go to the area where Hemlock Woolly Adelgid is native, and you find the biological control agents that are abundant and affect important controls in the area where they're native, and you evaluate them to be sure that they feed only on the prey items, the items the insects that you're trying to control, and then you bring them over. So there's basically two approaches to implementing the classic biological control. You can wild collect predators in the area where they're native and bring them back and then directly release them. This is relatively inexpensive. You just need to go over there and collect and bring them back. But there's always an inconsistent supply of predators. Availability fluctuates in the wild native populations. And there's also the possibility of doing this on the East Coast, where the predators have been present and populations have been building. We've actually gone down to areas in North Carolina and Maryland, and perhaps also in the Delaware Water Gap, and collect predators there, and then we can distribute them from that point. The other method is to lab rear them in an insectary, and the beauty in this is that it's more or less a consistent supply of predators, knock on wood there, and it's scalable. Basically, you can scale up the laboratory and can get more insects. It's a relatively expensive way to get insects, but really that is the point we are at right now, is that we need bugs, and we need them as fast and furiously as possible, because the numbers of adelgids out there are huge, but the numbers of predators we release are tiny, and we don't really have a long time we need the populations of the predators to build as rapidly as possible. And our hope is that we can do that before the populations really get into the heavy, dense populations of hemlocks in the Adirondacks. If we get the populations of predators built, then to the point where we can have a lot to release and get some kind of control, excuse me, by the time they get into the Adirondacks, we'll be in good stead. The labs have been very successful. Virginia Tech in Georgia and in Tennessee at growing the predators, and we're actually very fortunate to have the experience of our colleagues down there as we start our own lab up here in Ithaca. So when thinking about the biological control of HWA, there's basically two different guilds or feeding guilds, right, of the predators. There's the winter-spring feeders, which basically start feeding on the cystins generation. Excuse me, hold on. And then there's the spring-summer generation, which begins feeding on the eggs of the progrediens generation, and then on the eggs of the cystins generation before they settle down and go into estivation. So there are these two groups. There's different insects involved. So the spring-summer feeders, we have the beetles, Sasajiscymnus. It's a ladybird beetle, a coccinellid, Scymnus, another coccinellid, and then Leucopis, which is a silver fly. And I'm going to talk about all these after this. And the winter-spring feeders, basically the only one we've been working with now is the beetle Laricobius. It's a little deodontid beetle there. So the classic biological control program for Hemlock Woolly Adelgid basically started in the early 90s with the importation of Sasajiscymnus tsugae, which was brought in from Japan. And after that, there was exploration in China, in Sichuan province in the mountains where the hemlocks are. And there were a number of species that were brought over. Actually, it's an interesting story. They're basically the Scymnus, or the subgenus of the Scymnus, the Neopolis, were basically unknown, or not unknown, but they're, after the work looking at the predators of the Hemlock Woolly Adelgid in China, they basically doubled the number of known species in this genus, or this subgenus there. Interestingly enough, in China, they found very few Laricobius. It's mostly Scymnus, whereas the Laricobius nigrinus that we've been working with comes from the Pacific Northwest. It was brought over from actually a seed orchard, a hemlock seed orchard in southern Vancouver Island by Lee Humble and Scott Salem, and they've been working with it there at Virginia Tech for a while. And then Laricobius osakensis from Japan was brought over in 2005, and that's sort of a recent bug. And now Leucopis species from the Pacific Northwest. I'm going to cover all these in greater detail here. So this is a picture of all the releases to date. This is a heat map. I know that there's more releases in there that aren't on there, but this gives you a good idea. You can see where all the major effort has been down here in North Carolina, Great Smoky Mountain National Park, and Tennessee. And and in the Delaware Water Gap, this is another pretty map. I like these things. I like maps anyway. So it gives you an idea. So they're spread out pretty much all up and down the coast, up all the way up into Maine. The different species are released in different locations. So Sasajiscymnus tsugae, basically it's the coccinellid. It's a spring-summer feeder. First brought over in '92. Mark McClure brought it over and worked with it in Connecticut, and they were growing it at a number of locations: New Jersey, Tennessee, North Carolina, South Carolina. Relatively easy to mass rear. The first releases began in '95, and it's interesting that in excess of 5 million have been released to date. And this is basically where they've been released. This is up here in the Delaware Water Gap. All the way up into Maine here, and then down in this area. The sad thing is that 862 releases since 1995 is that recoveries have been very low. and indeed, those in New York, I've never found it at any of the release sites in New York. And down here in the South, they have similar problems finding it. My colleagues, Pat Parkman in Tennessee, thinks that it's definitely better, it does better in the more southern regions where it's warmer. And so I'm just, I don't know, we're basically, the book's out on it. We're not really sure what's happening, but the research emphasis has shifted from Sasajiscymnus tsugae because of the lack of recoveries and to other species at this time. So the Scymnus species that were found in China were brought over by Mike Montgomery, and they focused on 3 species at that time. They were unknown at the time. They're all abundant predators in China, but the problem with Scymnus is that they're very difficult to rear in the lab. 2 of the species were grown, Ningxinensis and Senulodus, forget it, you can read that. They were released in 2004, but they were released in relatively limited numbers, and no recoveries have been made really of them to date. Scymnus camptodromus, on the other hand, looked to be very, very good predator. I know Melody Keena was very, very keen on that, and so just when they were doing the work, the colony, they had the colony at Penn State, and the NAPO review required another test, and the colony just collapsed at that point in time. Couldn't get it out in time. So this is a bug that really shows great promise, but it's stuck over in China now. We don't have a colony, but it's there. It's something that we're looking at, and it might actually be a good thing, because I think we really need a spring-summer feeder that is effective. I think we need a suite of predators if we're going to really affect biological control. So this is Laricobius nigrinus. So it's in the family Dirodontidae. It's a winter-spring feeder. So basically the pupae emerge from the soil in the fall. The adults then the adults emerge from the soil in the fall. The adults then feed on the developing nymphs of the cystins generation through the wintertime. And then when the HWA lays their eggs in springtime, that's when Laricobius lays their eggs in the springtime. And the Laricobius larvae feed on the progrediens' eggs, and then they drop into the soil to pupate. The important thing here is that they feed only on the progrediens' eggs. They don't feed on the cystins' eggs. So there's a window there of opportunity to get to slide in other predators. So the Scott Salem and Lee Humble brought it over in '97 and did a lot of work on it. The first releases were made in 2003 down there in North Carolina. And the labs have been successfully rearing this insect in Virginia, Tennessee, Georgia, and now we have our own lab colony going here in New York. So to date, over 330,000 have been released throughout the eastern states, all the way from Georgia up into New York and Massachusetts, and I believe New Hampshire as well. Establishment has been widespread. And as perhaps with Sasajiscymnus, the establishment appears to be better in warmer areas. I know the population growth hasn't been as dramatic up here in New York. I've been releasing it since 2008, and I've yet to see populations build like they have in the South. So this is the Laricobius nigrinus release sites all up and down the coast, and you see the preponderance of releases down here in the South in North Carolina and Georgia. The release sites in New York, I've released since 2000, actually it was fall 2008, at 19 locations. And there are 2 biotypes. There's a Puget Sound biotype, and then there's an interior Idaho biotype. And we feel that the Idaho biotype might be more cold tolerant. But right now, I'm sort of undecided. I found both of them to be established in New York State, and I don't know which one actually might have the populations be building more rapidly. We really need to do more work on that. Here is the lab that we have. We just started. pay no attention to the picture in the bottom right. Goofy guy in the costume, that's me. But anyway, so this is the way we're rearing the Laricobius in our lab, and right now we are just beginning to get Laricobius eggs, and hopefully we'll get we'll get the larvae dropping down and we'll get pupation shortly. We hope to do our first releases from our lab here in this fall. And so Laricobius osakensis is another one that was found in 2005. It was first brought over, and it was brought over from southern Japan, where the HWA from on the East Coast comes from. They've been growing it at Virginia Tech, and their research shows that it might be more fecund and more voracious than Nigrinus, but we're not really sure. And we have a little bit up here, we're trying to grow it, and so we're very hopeful that having another Laricobius might actually help out the Laricobius nigrinus in reducing the populations. The big hope that I have right now is with Leucopis, the silverflies. There are two species of silverflies, the family Camimyidae. They are spring-summer feeders. Now, imagine you have Laricobius, they feed over the winter, and as I mentioned earlier, you get that tremendous amount of mortality of the cystins generation in the winter. Well, when the Leucopis come out and they feed, they lay their eggs in the eggs of the developing Adelgids. So they're basically, the carpet is not pulled out from under them. They're laying their eggs where there are Adelgids, and those Adelgids won't be dying. So I think that this has a definite advantage in the north as opposed to in the south, where it might not be as much as an advantage. So this actually is a very important predator in the Pacific Northwest. Joe Ross, Kimberly Whalen, and Glenn Kohler have been looking at these and really recognized their importance in the 2000s. And the first work with them really on the East Coast started in 2015. The work showed that it can become established both down in the South in North Carolina and up here in New York. And I did a couple of releases in 2015, but really we started releasing it in New York in numbers in 2017, just this last spring. And we released it at 10 locations, and at each one of these locations we got establishments. So everything looks pretty good right now, but we have to follow them through the winter time to see how they're handling the winter. There's a picture of the egg, and amongst the egg mass of the HWA, there's a larvae feeding away. They like to burrow in there, and they'll just pop those eggs and suck the juice out of them. And there's a pupae right here. You can see the pupal cell, and this is a newly emerged adult. And this is all from our lab work here at the Sakarya Arthropod Research Laboratory. This is a quarantine facility. So the reason we have to use a quarantine facility is because we need to bring these insects over as larvae and then rear them out to adults, and we don't want to, when we bring them over as larvae, they're in amongst the Hemlock Woolly Adelgid foliage from the Pacific Northwest. And I'm not going to be the one to introduce the Western clone of Hemlock Woolly Adelgid to the East Coast. And so in the quarantine facility, we put the foliage in these plexiglass cages with mesh net on them that's it's 120 microns, so it's half the size of any crawlers. So no crawlers will get out of these cages. And we rear out the adults. And because we don't want to work with the larvae, because the larvae have their own parasitoids, so we don't want to introduce those parasitoids on the East Coast. Here are the adults. And one of the reasons that it's important to bring them to the East Coast to work with is that we're finding that the adults are actually quite short-lived. We did a short experiment this spring, and we found that over 50% of the population of emerging adults was dead after just a couple of days. And so we're working to try and figure out how to minimize that amount of mortality. But in the meantime, we want to get them out, rear them out, and then release them as quickly as possible. And we do that in bags like this, where we actually put a bag on the tree, and then we put the flies into the bag. We put about 5 males and 5 females in each bag, sometimes more, sometimes less. And then we go back, and after that generation has a chance to establish itself, they'll lay their eggs and they'll begin to develop. Then we take the bags off so that those progeny can then disperse naturally. The cool thing is that there's probably 2 or maybe even 3 generations of this insect throughout the lifespan of the eggs of the progrediens and the cystins generation. So we get multiple generations, and without that die-off of the prey, I have hopes that this might be the answer in the north to our Adelgid biocontrol. So this is just the numbers. I brought about 80 branches shipped to the quarantine in May. We reared out a total of 4,400, almost 4,500 Leucopis adults. We sent some to South Carolina for Bud Mayfield to use, and we released here in New York almost 1,700 during May and June. And we found the F1 generation at all release sites. So stay tuned with this one. But this is sort of a placeholder just to remind you that, sure, Mike Montgomery did exploration here in China. There's been some work done here in Japan. But there's still a lot of exploration to be done for predators of the Hemlock Woolly Adelgid, in particular the Pacific Northwest. Hemlock goes all the way down into California, into the Sierras. It's here in the Rocky Mountains where there's Adelgid populations. Hopefully, maybe where the predators are more cold adapted. So that might be a really important thing to look at, as well as further up here along the coast. So there's still a lot of exploration for natural enemies to go on, and we don't know what we'll find. But I think that it's important to go in that direction at this time. In the meantime, we have valuable tools in the systemic insecticides, and we've been using them in New York State to save some of our valuable scenic and genetic resources. There's many state parks right now that would really have hugely diminished populations of hemlocks if we hadn't treated them. We use a cocktail, a tank mix of imidacloprid and dinotefuran, and the imidacloprid is great because it's effective for 7 years or more. The problem with it is that it's slow to move through the tree, so sometimes when you have a big tree that's already impacted by the Adelgid, it might not have the capacity to move it up into the tree and up into the canopy. So we use dinotefuran, which is very fast to move into the tree, takes down the populations, retains the canopy so that it can then move the imidacloprid up into the tree. And so in New York State, basically for best management practices, we've been doing a basal bark spray, which we treat just the basically 5 foot of the tree with the insecticide and just until the point where it's just about to drip down, and then the tree takes it in through the bark and moves it up into the canopy. It's been very effective, and the efficacy, how long it remains effective, I'm not really sure. The efficacy for the basal, soil treatments has been shown to be where you put it directly into the soil, either inject it or pour it onto the soil, has been shown to be about 7 years. We're not sure if the basal bark spray will be that long, but we're watching it right now. We're right at that cusp of about 5 years. So stay tuned on that account. And this is one of the instances that we're using it. This is actually out in Zoar Valley, which is south of Buffalo, where we have a huge stand here. You can see this is a Nature Conservancy preserve where we have about 300 plus acres of old-growth hemlock. And I went out there a few years ago with the steward, and sure enough, we parked here, and then we walked up the creek, and there it was, Hemlock Woolly Adelgid on one of the trees. Well, subsequent investigations that fall showed that it was just located in that one area. And so we located the trees, we treated the trees, and we treated a buffer all around them. And we have yet to find Hemlock Woolly Adelgid back in that stand. Is it gone? I don't really know. But this is an effective treatment, and this is exactly what we did up in the Adirondacks this last fall, when we found it at Prospect Mountain, where there were only 3 trees that we found infested, and we treated a whole buffer around those 3 trees, and hopefully we'll basically stave off population growth of Hemlock Woolly Adelgid until we can hopefully have time to get the biological controls in place. But this is a very isolated stand. This is really the only area, one of the few areas in western New York where we have where we found Hemlock Woolly Adelgid. I sort of wonder if it's nearby. I don't really know. It seems like sort of a miracle, but we'll use it. No worries. And lastly, I wanted to bring up some silvicultural approaches to controlling Hemlock Woolly Adelgid that have recently been brought. They're being tested by Bud Mayfield from the Forest Service and Bob Jetton from NC State. And it's based on work that shows that the estivating systems in the summertime are sensitive to direct light. I've been doing some shade cloth experiments here in Ithaca, and work has also been done in North Carolina that shows that indeed when you shade the trees, there's more survival of the estivating systems. So Bud and Robert Jetton are basically getting into stands where they're basically hardwood stands, not dense hemlock stands, but hardwood stands with hemlocks in them, and they're thinning out the hardwoods to open up the canopy, and they're looking to see if that indeed causes mortality of the Hemlock Woolly Adelgid enough to keep the trees alive. So this is a short commercial plug for, not commercial plug, the Hemlock, New York State Hemlock Initiative is my group here at Ithaca at Cornell University. Basically, we're a group that we work with outreach and trying to help various members, various constituents within the state get together to implement management of the Hemlock Woolly Adelgid in the hemlock forests. And it also includes the biological control program here. So our website, I don't know if you can see it at the bottom there, it's nyshemlockinitiative.info. You can go there and see all the stuff that we've been doing. It keeps changing rapidly. Got a good crew here. I'm very, very proud of what we've been able to do. There's that bucolic photo there. I have to thank Cornell College of Agriculture and Life Sciences for being so patient with me over the years, and then funding from the state Department of Environmental Conservation, the Forest Service, and then APHIS. So with that, I will take questions, which I have not been paying attention to at all. So Peter, perhaps you can help me.
Yes, Mark, happy to. So there's just one question that's come in so far. So this is a chance to type in questions. And before you do that, let me just say, please send it to all panelists and attendees. It defaults to just all panelists. So send it to everybody, panelists and attendees, so everyone can see the same questions. For those of you who are interested in continuing education credits, you've done all that you need to do via your registration. So you don't need to do anything more. I will process that. It's a manual processing, so I just have to find the time to do it. It usually takes me a couple of days, and I'll send that out to you all. So if you have questions, please type them in for Mark. And there is a question from, I gotta put my glasses on, I can't see these small typesets. Irene says, what are the native natural predators of the Adelgid in the Northwest? So did you cover that, or you want to do a rehash?
Well, it's actually interesting. No, the important ones are the ones that we're working with right now. There's Glenn Kohler did a great study of the diversity of predators out there, and there's over 50 species that they found associated with the Adelgid. A lot of those are generalist predators, and of course for biological control we do not work with generalist predators. We work only with specialists that feed only on the prey item that we're looking at. The two most important predators in my mind are, I think, the Laricobius nigrinus, and then the silver flies, the Leucopis, argenticollis, and piniperdo. I was really floored at how many Leucopis that I was able to get out of seemingly a small amount of foliage that I shipped back. So there are probably others that we could look at. I don't know what they are. I think we need to do more exploration, perhaps in colder areas and different climates other than just the Puget Sound area.
Okay. Here's a question, whether or not low temperatures also reduce, scrolled off the page, low temperatures also reduce predator populations.
That is something that is on my agenda. I haven't been wanting to freeze bugs. I'd rather release them, but indeed, we're going to start experiments looking at the cold tolerance of the predators that we're working with. We have a great facility here at Cornell that it's funny when you go to a college town and you go to a party and you start talking to people and they say, oh heck, you've been using an antiquated machine. You should try our machine here in the engineering school. And they got a great machine for freezing bugs. Of course, they never thought of freezing bugs in it, but we've been using it, and it's come up with some really interesting data that I didn't have time to present today. But
okay, then there's a question that's kind of a two-part. One says, how much do the predators cost? And somewhere below, I guess same person says, can private landowners buy the predators?
No, that's a good question. I don't know how much they cost. This is our first year of producing them here. My colleagues at other labs feel like they might be like, more than $5 each, $8 each. It's really hard to calculate that out. But so far, all of the labs that are growing these predators are supported by public funding, and they're not available to private landowners to purchase. There is one lab that produces Sasajiscymnus tsugae in Pennsylvania, and they have a web presence, and they're trying to sell them. I discourage anyone from buying them. I think it's a waste of money because we have yet to find them to be effectively established and perhaps controlling HWA populations. I think that at this time, if it was me and my land and I had a bunch of hemlocks and I got Hemlock Woolly Adelgid, I don't think the predators are going to be able to build their populations enough. I would get into using the chemicals, do a triage of your trees, choose which ones you wanted to save because of their genetic beauty or whatever, and work on that until we can actually get better populations of the predators going.
Okay. And speaking of insecticides, we had a presentation about a year ago by Elizabeth Benton, and I posted a link to that webinar in the chat window.
Oh yeah, great. Elizabeth Benton's work is fabulous. She's really, really good on Hemlock Woolly Adelgid.
So does the size of tree have any impact on whether or not, or the extent to which there's an infestation?
No, I don't think the size of tree matters really. The tree, the thing that matters is how long it's going to be able to survive an infestation by Hemlock Woolly Adelgid. It's just like with any silvicultural system. When you have a tree that is healthy and robust, and it's going to survive longer than a tree that might be, I hate to say, but over mature or huge with a slow vascular system. Or perhaps one that's suppressed. In those instances, those trees are going to decline more rapidly than one that's been maintained with a full canopy with a really large crown. Yeah.
Are there any chemical sprays that you can put on the hemlock seedlings to protect them? So a preventative rather than a reactive treatment?
Well, the systemic insecticides, imidacloprid and dinotefuran, they will work. Dinotefuran will persist for a couple of years, whereas imidacloprid will persist for a lot longer. I quite frankly, I don't advocate prophylactic treatment because you should treat when you get the Adelgid, and that's sufficient.
Okay. To what extent do trees recover after a winter where there's high Adelgid mortality?
It's very much the same reaction that I see when we treat the trees with insecticides. They can mobilize the resources and put out more new shoots. Those shoots tend to appear at the top of the tree. So it's really important when you're looking to see if your tree is in trouble. I forgot to mention this. The best time of year to look at the health of your tree is in springtime, when those nice bright green shoots are coming out. If your tree is infested and you don't see any of those shoots, or you see very few of those shoots coming out, that's when you should be really concerned, because the buds are not producing new shoots. And that's consequently the way you can tell if it's been effective, where you look at the tree, and if it has lots of those nice bright green shoots coming out of them, as opposed to the dark green older foliage, it just stands out like a sore thumb, and you know that your tree is doing well, putting out those new shoots.
Okay. Two different people asked whether or not birds are a control mechanism of Hemlock Woolly Adelgid.
You know, it seems like a no-brainer that there's food on there, and lots of it, but I've been looking at a lot of Adelgids for many years, and I have yet to really see birds feed on them. You know, maybe that white woolly stuff is a yucky thing for them. I have no idea.
Have you tried them?
No, I have not. I've tried bark beetles. They're really great. They have that real piney flavor, you know, when they come out of spruce. A little bit of garlic. These have
a hemlocky flavor. Okay, on a serious side, back to the questions.
No.
Are you finding black scale along with Hemlock Woolly Adelgid, and what thoughts do you have on its control?
Black scale, I think you might be thinking of elongate hemlock scale. And yes, elongate hemlock scale is present. It's interesting. It hasn't moved as rapidly as Hemlock Woolly Adelgid across the landscape. It was introduced in the early 1900s in the Bronx Botanical Garden there, or now the New York Botanical Garden. And it's been spreading. It's very common in the Hudson Valley, and now I think through the movement of nursery stock, it's found in a number of places throughout New York State. The cool thing is that the treatment with dinotefuran is very effective against it. And so that's one of the reasons we like to use dinotefuran in combination with imidacloprid for the treatment of Hemlock Woolly Adelgid. The imidacloprid itself is not effective with elongate hemlock scale. You have to have the dinotefuran. There has been one predator of the elongate hemlock scale, Sasajiscymnus, that it's been found. It was actually introduced for another scale insect, but I have not seen that grow in numbers nor be effective. Although I think it would be a cool thing to look at Sasajiscymnus and perhaps other predators of the elongate hemlock scale as well.
Okay. And there's a couple of questions about insecticides. There's one about environmental hazards, and I'll just refer that question back to the link to the webinar that Elizabeth Benton did, which was all about the the risk analysis associated with herbicides. So I'm sorry, insecticides. Yes, thank you. So question is kind of the survival strategy of the predators in the Pacific Northwest. So if there's, will they flip from Adelgid to other species? So if there's a lull in the population of the Adelgids, do they flip to another species, or they just jump from, one outbreak area to another outbreak area of Adelgids? So what's their flexibility?
No, they're not flexible at all. That's why we work with them. They only feed on Hemlock Woolly Adelgid.
Okay, and can you use biocontrols in conjunction with insecticides?
Yes, you can use biocontrol in conjunction with insecticides, because you just can't treat all the trees out there. So in many of the state parks where we've been treating the trees, we treat the biggest, the most beautiful trees, and we let a certain number of the trees go untreated to harbor the Adelgids, and then we release the predators at the same time, so they can feed on the untreated trees, the Adelgids on the untreated trees.
Okay. And then what's been the survival of hemlock in Watkins Glen State Park?
They look pretty good. They were treated right at the last minute. Some of them are looking really bad, but they appear to really be coming back. The interesting thing about the treatment I wanted to mention also is that the canopies were already pretty diminished, and the lower branches seem to die off first, which sort of makes sense, because that's where the epicormic buds are not going to form. They're going to form up in the top of the canopy. So when they get get treated, it's actually, you see the response not down low, but up in the top of the crown. That's where all the respiration is going on. And so when you look at the trees, in fact, there's been a couple instances where applicators have actually come to me and said, Mark, I'm freaking out. My trees aren't recovering. And I went and visited the site, and I said, well, you just got to look up in the top of the tree. Look for those nice, bright green, expanding shoots up at the top of the tree, and you can see that they're doing really well.
Okay. It's all the questions that I see. So, Mark, I want to thank you for taking time today to put this presentation together and to talk with over 100. We had 109 participants, and I had at least three people could knock it in. Others maybe just didn't respond. But so I'm looking forward to another good turnout at the evening session. So thank you, Mark, and I hope you all have all the participants